everyone, welcome back. Today's video will be about titrations of amino acids. This video will discuss the following topics. What is a titration? What are the types of titrations? Why do we titrate amino acids? How are amino acids titrated? An example of an amino acid titration. And what is the isoelectric point? It is also important to note that throughout this entire video, I will explain how to understand and interpret a titration curve. As always, if you want to skip to a specific section of this video, you can do so by clicking the relevant timestamps found in the description section below. Also, just a quick reminder, please make sure to subscribe to the channel as it helps me a lot. Currently, only 1% of my viewers have subscribed to the channel. All right, let's get into the video. What is a titration? A titration is a lab technique used to determine the concentration of a solution with the reagent of known volume and concentration. It can also be used to graph a titration curve, which can then be used to determine specific values, as you will see in today's example. Titrations often use the following setup. The titrant is the reagent with the known volume and concentration, and the titrand, that is ending with a D, is a solution with known volume but unknown concentration. The titrand, ending with a D, is also called an analyte. Types of titrations. These include but are not limited to acid-base titrations, redox titrations, and precipitation titration. Amino acid titrations fall under the category of acid-base titrations. In these types of titrations, we can use acids as the titrand and a base as a titrand or an analyte, or vice versa, meaning that you can use a base as the titrand and an acid as the titrand or the analyte. Why do we titrate amino acids? Titrating an amino acid allows us to determine the titration curve for the amino acid. The titration curve can help us to visualize the acid-base behavior of an amino acid, and it helps us to determine the pKa values of the ionizable groups of the amino acid. If you want to learn more about the acid-base chemistry of amino acids and pKas, click on the card on the right-hand top corner. How are amino acids titrated? Let's take glycine as an example. Glycine is a simple amino acid. The four groups that are attached to the central carbon atom of glycine are the amino group, the carboxyl group, and two hydrogen atoms. One of the hydrogen atoms is the R group for glycine, and the other hydrogen atom is found in all amino acids. The amino and the carboxyl groups of glycine are what makes it an ideal candidate for titrations. This is because the amino group is basic and the carboxyl group is acidic in nature. So here's an example of a titration. Say we decide to titrate an acidic solution of glycine with 1 molar concentration. In acidic environments, glycine would exist as NH3+, CH2, COOH. This is because in acidic conditions, proton ions are abundantly available and protonate all ionizable groups. Therefore, the glycine molecule in acidic conditions would be fully protonated and would have an overall +1 charge. We would then titrate this solution with a strong base like NaOH. During this titration, it is important to document two things. Firstly, the amount of NaOH being added and the pH of the solution. Once everything is set up, the titration begins. As the acidic glycine solution is being titrated with NaOH, the carboxyl group will begin to deprotonate first. This is because the carboxyl group is acidic compared to the amino group, and we know that acids are compounds that give up their proton or H+ ions very easily. When we have added 0.5 equivalents of NaOH to the titrand, that is the acidic glycine solution, we create a solution with equal concentrations of glycine and its Zwitter ion. That is, we have equal concentrations of NH3 plus CH2 COOH and NH3 plus CH2 COO minus. There are two important things to note here. Firstly, this is the point at which we can determine the pKa of the carboxyl group. The pH at which the concentrations of the protonated glycine and its Zwitter ion are equal is the pH at which you can determine the pKa of the carboxyl group. In other words, the pKa of the carboxyl group is the pH of the solution when 0.5 equivalents of NaOH is added to the glycine solution. Secondly, if you keep adding more NaOH to the glycine solution, you will find that the pH reading does not dramatically change as it did before. This is because the glycine and the Zwitter ion solution now behaves as a buffer. You can see this in the titration curve. This is where the curve is relatively flat. Just as a quick refresher, a buffer is a solution that resists pH changes because it neutralizes small amounts of acids and bases. Buffers can do this because they consist of equal concentrations of a weak acid and its conjugate base. In this case, the weak acid is glycine and its conjugate base is the Zwitter ion. As we keep adding more NaOH, the carboxyl groups of glycine goes from being half deprotonated to fully deprotonated. All glycine molecules now would exist as NH3 plus, CH2, COO minus, and therefore the glycine solution is no longer a buffer anymore. If you continue graphing the titration curve at this point, you will see a rapid increase in pH of the glycine solution. When we add exactly one equivalent of NaOH to the titrand, that is the glycine solution, we would have a solution of glycine where all glycine molecules exist in their Zwitter ion form. This is because we began with 1 molar of glycine solution, and therefore we had 1 equivalence of glycine to begin with as well. This also means that in that 1 molar glycine solution, we had 1 molar carboxyl groups and 1 molar amino groups. When we add 1 equivalence of NaOH, 1 equivalent of carboxyl groups became deprotonated. This meant that all the carboxyl groups available were deprotonated. Because all glycine molecules are now at the Zwitter ion stage, all glycine molecules are also electrically neutral. Because of this, the pH of the solution is now equal to the isoelectric point of glycine. Therefore, another advantage of graphing a titration curve when it comes to amino acids is identifying not only the pKa of the carboxyl groups, but also the isoelectric point of glycine. What is the isoelectric point? Just as a quick refresher, the isoelectric point is depicted by the symbol Pi. It is also defined as the pH at which a molecule is electrically neutral. For amino acids that do not have an ionizable R group, the isoelectric point can be calculated using the following equation. Pi equals the sum of the pKa of the carboxyl group and the amino group divided by 2. This also means that the isoelectric point for all amino acids that do not have an ionizable group is the same. When the titrand, that is the glycine solution, has reached its isoelectric point, it is extremely sensitive to pH changes. That is why at this stage the graph appears almost vertical. As you keep adding more NaOH, glycine enters the second buffer stage. This is because the protonated amino group now starts to become deprotonated. When the concentration of NH3 plus CH2 COO- is equal to the concentration of NH2 CH2 COO-, the glycine solution now acts as a buffer again and has entered the second buffer stage. Similar to the first buffer stage, this is the point at which we determine the pKa of the amino group, the pH at which the concentration of NH3 plus CH2 COO- is equal to the concentration of NH2 CH2 COO- is the pKa of the amino group. In other words, the pKa of the amino group is the pH of the solution when 1.5 equivalents of NaOH is added to the glycine solution. At this stage, the titration curve will once again look flat, depicting the second buffer stage. As we keep adding more NaOH, more glycine molecules in their zwitterion form will become deprotonated. When exactly 2 equivalents of NaOH is added to the glycine solution, all glycine molecules will have become deprotonated. All glycine molecules will be negatively charged with the overall structure of NH2 CH2 COO-. And that's it for this video. If it was helpful for you, please make sure to subscribe to the channel. And that's it. I'll see you next time. Bye.